Welcome to Desde el Origen. My name is Dr. Carmen Maldonado Blar, your host, and today we have a wonderful guest on our podcast, Dr. Filipa Godoy. And I'll tell you a little bit about our special guest in our podcast. Dr. Godoy is originally from Portugal, hence the wonderful outfit that she has on. And I'm also going to mention my Camino Portugués, which I did a year ago, almost a year ago, which was wonderful. And Filipa is a microbiome researcher who has specialized in the field of more than 20 years. I mean, that has been her passion. And when we talk about her background, scientific background, we see that she started as a plant biologist after she receiving the Erasmus Fellowship in Spain. And then in 2003, she came to Puerto Rico planning to study tropical biology. However, she met her now important mentor and very important guidance for her as a scientist, Dr. Gloria Domínguez Bello, who was a colleague of mine at the Department of Biology. And then with working in this laboratory with Dr. Domínguez Bello, Filipa got very much involved what soon to be is called now microbiota field, that in the past it had not had such much interest, but now it created this niche. And she finished her PhD with us at the Department of Biology, and later she continued postdoctoral work. And right now, Dr. Godoy leads a pioneering research while being chair of the Department of Microbiology and Immunology at the University of Puerto Rico School of Medicine. And in addition to being a scientist and an administrator, she's also the PI of a very important institutional grant that we'll be talking later about, the COBRE grant, and that is a Center for Microbiomes Sciences, where she supports research, fosters collaborations, and helps shape the next generation of microbiome scientists across the Caribbean. And it's fair to say that Dr. Godoy brings with her an incredible diversity of insight, curiosity, and experience. And we're so excited to really get and talk to her about her career and her plans and also her passions. Welcome, Filipa.
Thank you so much.
To Desde el Origen.
I'm very humbled, I'm very honored, and very happy to be with you.
Thank you, Filipa. I have to say, on a personal note, I know Filipa for so many years because when she was our doctoral student, her lab was right next to mine. And so through the years, I knew her development. When she defended her thesis, I was there. I remember Maria Gloria being so proud of Filipa. And to this day, that happens. So, and also the work that Maria Gloria Domínguez pioneered is something that really revolutionized the field. Mm-hmm. And it just happens that Filipa was part of that. Of course, when we have students, and Filipa, now that she trains students, know that the work that is done in the lab, the PI always, gets the inside ideas, but at the end of the day, it's the students that really do the work and that inspire us as well as professors and mentors. That's true. So I want to give kudos to Maria Gloria.
For sure. And I still collaborate with her.
I bet.
We have a bunch of projects. She's on thesis of students in my lab. Mm-hmm. We have new projects, papers coming out. Yeah, and she's a friend. So we have developed both a professional collaboration, but we keep as friends.
Yes. But that is
through the years.
I think that is one of the a part of science, when we are as scientists, and if we have a good relationship with our mentors, there for a lifetime. I mean, they
if it's based on loyalty, trust,
Yeah.
It's forever.
Yeah, my first students, I still keep in touch with them. They always reach out to me when they need help or.
Definitely. I'm always calling her. I have a situation, even administratively, because she's senior. I'm like, how do you see this? Do you think I should you always reach out.
Yeah, I think it's a very special relationship. So now we'll talk a little bit more in detail about you. Okay. But before we talk about your projects and the way you achieve what you have done thus far, we usually, for every guest of our podcast, we ask them, what was the experience at the beginning when you were young that really triggered this interest of you as a scientist? If there is any, or a group of memories that you would like to share with us.
I would like to share. So I grew up in a place called Cartacho. It's in the center of Portugal, north of Lisbon, half an hour north. And it's a rural area. My father worked at the big company in Lisbon, and my mother educated me. She was working, and then she left her job while I was a teenager to guide me and to be with me. And my father was enamored of. He's a naturalist. So he has a museum with stones, and he loves geology, loves natural history, and so I grew up in a rural area, but with he has like a certificate of National Geographic since the 1960s.
Wow.
So
okay.
I grew up, with these magazines and all the time, and Geo, which is a magazine from France about natural life. And I was in love with those, with the pictures and with the images. I think those are the first memories of how exciting it would be to work with nature. So at some point I wanted to be a photographer for National Geographic, especially underwater. I loved marine life, I loved plants. So I always wanted to be, I knew I wanted to do something related to biology, but even in the 12th grade I was not sure if I was going to become a biologist or not. But I chose biology because it was the thing that attracted me the most. But I was not in love with the field. And during the years, I had very good professors, especially in forestry. I followed in Oporto. We had, I don't know now, but at the time, we had the scientific degree, which is plant cell biology. Then we have the teaching degree, if you want to go to be a teacher in biology. And then you have a molecular degree. I chose the plant cell and ecology side. And with that, I asked for the Erasmus, and with the support of a microbiology professor, with which I'm still in contact for 30 years. And I published papers with her after I graduated from here, from my PhD. In fact, we met in a meeting in Australia, and I'm like, you were my teacher in undergrad. I love you so much. And then we got in touch again. And through her, I got the Erasmus. She sent me a letter of support, and I went to. With Erasmus, you can choose. They give you a map, basically, and where do you want to go? Iceland, Spain, Italy. And I said, I don't know Spanish, and I never lived in an island. So let's see. Let's go to Canary Islands. And I chose the Canary Islands because they have a very good degree in algae, algology. So I worked with cyanobacteria, with their effects in tomato plants. Again, the toxicity of, developing new ways of, growing tomato plants. So I worked with that and another bunch of side projects that they had. And in that time, I met my soon-to-be husband, who is Puerto Rican. And I continued my passion for, biology and plants. I was in the beach all the time collecting algae. So I knew I wanted to do something about that. So when I met Maria Gloria here, the first meeting, she had an empty lab. She had a table, and she was on her computer writing something. And I introduced myself to her, and she said, oh, you're a new student. I just came, I don't have anything, but I can tell you about my studies. I work with Helicobacter pylori, and I was like, I don't want clinical stuff. Do you work with plants? I want to work with enzymes, lysozymes, especially lysozymes. And she said, well, that's nice. Do you know that there's lysozymes in animals? We could do that. And I'm like, And then she told me about all this Hoatzin work that she started, and was the cover of Science when they discovered that this bird could work like a cow physiologically. And I was like, wow, I want to be part of that project. And so I fell in love with the Hoatzin, and then the story goes on.
Yes.
So my memories come from, my childhood in Cartaxu with my parents. We would go to the beach on weekends. It's a very quiet town, small town life. And the magazines around me and, the experiences around me inspired me. But I think I am the first biologist in my family.
Okay, yeah. Okay.
Everyone is an economist, mathematics, most economists. Yeah.
Well, that's mathematics.
Mathematics. Yeah.
Yeah, but it's more toward the.
Yeah, I'm the first researcher actually.
Okay.
I have other engineers and stuff, but no biologists.
That's great. So, and for your parents, how was supporting you as a scientist? Did they approve that too, or they thought it was?
They approved.
Yeah.
But they always mentioned, it's hard. Science in Portugal, living with small grants, it's a hard, think about it. Maybe the private industry, but I was never interested in industry. So when I came to Puerto Rico, which is totally a different experience, because while I was an undergrad, I couldn't do research. I used to go to visit the labs and be with the grad students and wash their dishes and listen to the lab meetings, but the professor not even knew my name. It's totally different. So when I came here and I started, in the lab, I started working with people from your lab, from other people's lab, to go look for reagents. Oh, we need to do this PCR, and we need this and that. And people helped each other, and undergrads were, freely working with grads. And it's a new thing. It was a new thing for me. So I took advantage of that, and that helped because we didn't have any funds at the beginning. Yeah. Then she got the, the first funding.
The funding,
startup funding. Yeah. I don't know the name. Don't remember the name. And with that, we started, doing some work. And then I went to Venezuela with her to hunt the animals and to understand where they lived, how they raised the chicks. And then we developed the thesis, not only on the physiology of the animal, but also how they develop their families, how they groom the chicks, and how the microbiome is involved in this grooming and the development of the chicks into adults, which was very nice. And I had the chance, since we didn't have much funds, I had the chance to rotate in the US. So I worked at the Lawrence Berkeley National Lab, NYU, Washington University in St. Louis. I went to Australia, I went with NSF grants, and it was a very good experience. Doing the PhD in Puerto Rico, and that's something that people is like, oh, I did my PhD in Harvard you did in Puerto Rico, And what?
Yes.
I got 4 publications as first author, and like 6, in total I got 6 publications out of my work, and I wouldn't trade it because I'm in a tropical paradise. This is a lab. I live in a lab, in a tropical lab. So that was my experience.
Yeah, well, that sounds wonderful. And I think that it shows you that when you are connected with great science, it really doesn't matter where you do it. I mean, it's important. What really sets apart a project that could not be impactful is that you don't bring passion to it, and you don't have a mentor that really guides you.
Right.
And so talk to me a little bit about that mentorship relationship with Maria Gloria, which I hear, obviously, that was a wonderful experience. But where there's place where there was a lot of, communication between both of you about science, how she saw science and you see science during that time.
Yeah, we had different points of view, but we always talked about the things that we didn't agree on. For example, I think not only even weekly report. I mean, I reported weekly, and she met with me in person every week. But I think we were together daily. So she knew exactly the status of the project. Then other people started coming in, and I was like the technician that helped, get things together. So I was also reporting on other projects going on, and she involved me in those projects also to compensate a little bit for that effort. So I was involved in training students in analysis of what it was not microbiome, because we didn't have the pipelines that we have today.
No,
now we have time, and we have all these free open access pipelines. I had to go to the Max Planck to learn to code in Python for the analysis that I used to do in phylogenetics.
Wow.
Which would take like 5 days to run a phylogenetic tree with whole genomes of all these thousands of sequences that I generated, and now you can do it in an hour.
Yeah, it's
totally different.
I know.
But the time that she spent with me, I think is like 80% of the time.
So what would you think you have, let's say, incorporated from her style of mentor to what you are now as a scientist?
I think that I'm in control of my projects, but I also delegate in people I trust.
Good.
So that's the thing that I learned from her that I think is very valuable, because you give leeway for people to be empowered and to lead their projects, but they have accountability.
Yes.
And I know exactly the status of the project, where the samples are kept, where is the inventories, who is involved, what are the students. So everything is clear. And if from the start, from the get-go, everybody knows their responsibilities and who is in charge, and that you have to comply with this and that, everything is good. That's why I think I can handle 17 students at a time, because the ones that are senior, for example, my postdoc now runs basically the lab, and she reports back to me, this postbac is doing this, these undergrads are doing this. And okay, we have these 2 deadlines for these 2 papers, where are we? So we look at the data together. So that type of responsibilities that you share with people that are with you is important.
Yeah.
That you control it, but still you see how they develop. And that's why you can see everything from above, but you're seeing it all. I think that's the most valuable thing that I learned from her.
That sounds like a good thing to learn. And so here in Puerto Rico, when we think about microbiome, we think about Filipa Godoy, actually. I have to say that people are, you are the reference to go to. But maybe our audience doesn't know what really is the concept of microbiome.
Okay.
And so could you explain it in a way that everybody could understand what is a microbiome and why should we be studying this? Right.
So we know that microbes have been the first cells in the planet. The first. If it wasn't for cyanobacteria, there wouldn't be the ozone layer. They started producing oxygen. Everything that developed later, all multicellular organisms, evolved in presence of bacteria. So all living things are composed by bacteria, inside of their bodies and outside. So the microbiome is basically the composition of the communities that are associated with a host. And the host can be a human, can be a snail, can be a crab, or a sample of poop, a swab from an ear, samples from your eyes. Any niche, any sample can be an ecosystem to study a microbiome. And humans have microbiomes, distinct microbiomes across their bodies, because we have different canals. We have open canals like the oral cavity, the vagina, the anus, the ears, the eyes. You have different defenses, immune defenses, and proteins that kill your bacteria, like lysozymes, the ones that I wanted to study in plants. You actually have them, like eggs. Eggs have lysozymes that kill bacteria, because they kill specifically gram-positive bacteria that are environmental. We have lysozyme in our lágrimas,
tears
in our tears, in the cerumen of our ears. We have it in our vaginal tract to avoid infections from the outside. And then we have these microbial communities in all surfaces that talk to our bodies and by colonization resistance, which is a term that we use a lot, ecologists. It's colonize, the best guys colonize and, really take up the space, the habitat, so that the invaders cannot cross and cannot colonize. So that's why they matter, because we have 100 times more genes in our genomes that are from microbes that are from Homo sapiens. So for more than 500 million years, we have been co-evolving with these bugs. And we are, our health depends on them. And now we know that if you have a specific diet, you respond better to chemotherapy. If you have, there are treatments now based on the microbiome to treat disease, like Clostridium difficile, which is approved by the FDA. So the microbiome has come from being like this, microbiomania, it means nothing, it's just some sequences and some bacteria, to now, and really clinicians understanding that I need to know what's here to see if my medication is really working or not. On how can I modify the microbiome so that my patient responds better to this therapy, for example.
And so we could talk about health from the standpoint of how your microbiome interacts with the environment, of course.
Right.
No? So if there's deficits in the type of bacteria in the different areas of the body that you just mentioned, it could trigger situations of unhealthy settings.
Right.
So
we know that.
Can you talk a little bit about that?
Yeah. So the most diverse and the most known niche in the body is the gut.
Yes.
It has trillions of species. Then you have the oral cavity. Then you have the skin, which is a very large organ that is heavily colonized by fungi and by bacteria that protect our skin, the surfaces of our body. And then the least diverse is the vagina. And the vagina has very few species, like 17 species maybe. Depends on the groups of ethnicities, microbiome change with ethnicity as well. And in the case of the vagina, the fact that it's very low diverse, it's because you have hormones like estrogen that lead to the accumulation of sugar in the epithelium. And only lactic acid bacteria can use that sugar.
I see.
And by using that sugar, they ferment. And while fermenting, they produce lactic acid that drops the pH. And the low pH is a chemical barrier for the vagina. So one way that medical doctors have to know that if a woman has a vaginal infection is to look at the pH.
Yeah.
If you have a pH until like 5, you're okay. You have higher pH, it means that you have low lactobacillus.
Exactly.
Right? So that's one of the things. In terms of the gut, we could be here talking hours about the gut microbiome, because it has to do with obesity, it has to do with behavior, it has to do with response to treatment, it has to do with antibiotic effects of antibiotics and how do you respond to those antibiotics. I mean, the gut, there's a very common thing that we say about the gut is not Vegas. What happens in the gut goes everywhere.
It doesn't stay in Vegas.
Yeah, it doesn't stay. It goes everywhere. Even because in the gut you have bacteria like Bacteroidetes, which are very dominant and very important keystone bacteria that produces GABA.
Oh.
And
that's an inhibitor, right?
Yeah, so they even call it like an anxiolytic bacteria. So, there's already so much that we know about the microbiome that makes it now a true field that is important in medicine, and it's important in, for example, ecology. If you want to restore soil, and I was thinking about Elvira Cuevas.
Of course, that she studies the soil.
Yeah, she studies the soil, and she wants now to restore using microbiomes, communities, and see if productivity can improve food by improving the soil communities. So there's a bunch of applications of microbiome, not only in health, but I talk about mostly of health because of the School of Medicine and the context that I'm in, but we work with other things too.
Yeah, for example, from the neuroscience part, obviously, there's the brain-gut axis, and how through the vagus nerve, the microbiota from the gut is also regulating the brain cells. So it's just such an amazing field that truly has, for the last I think, 20 years transformed the way we see bacteria. And you were going to say something?
Yeah, we're going to say a little less.
Yeah.
Because the first paper, my first paper, it was the first paper on a bird microbiome. It was also the cover of the Environmental Microbiology. You remember?
We have that.
We had a lot of press. And it was 2008.
Oh,
so it's still
less.
It's still less. It's like, I don't know, 17 years, 15 years.
Almost, Filipa. We don't want to.
Yeah, we are getting old. Don't say that.
No, we are always getting wiser, honey.
Wiser,
not
wiser.
old.
Yeah,
No, never old. Wiser. So, so I think the microbiome is an excellent topic to study because of all the things that we just discussed. And so thinking about more of how environment and lifestyle can change, now coming from the outside, not talking about the inside, so how the external can change the microbiome. What is it known, or what do you think are the most challenging questions that this kind of interaction face?
Yeah, it's so true. Like we are at the level of, like, all humans share the same phylogenetic signal. One thing that I didn't say, microbiomes are transferred via mother to offspring.
Exactly.
In all mammals. So with the first contact that we have with microbiome is at birth. And that's the keystone work by Maria Gloria, was showing that babies who are born by a C-section have skin bacteria and environmental bacteria, while those who are born vaginally acquire the vaginal bacteria. And that's essential to understand then how the body develops their own bacteria. But one of the things that we also know is that depending on your sexual intercourse type, if you do oral sex, you will have a different oral microbiome than a person who doesn't do that. Depending on the diet that you have, it will change your microbiome. You don't have the same microbiome across the day because there are circadian rhythms, and sometimes you are dysbiotic in the morning, and then you have a very good lunch and you eat a lot of fiber, and then you're a little bit better in the afternoon. The built environment is also another field, and we are looking at that with Dr. Bolaños and with Dr. Cavalin in architecture, and Maria Gloria too, to look at how the microbiomes of homes impact the residents' health.
Wow. Okay.
And we've published several cool papers about that. And now Bolaños and I are looking at the impact of fungi from the environment with the fungi that live in our bodies. And because he has a station at the UPR.
Yeah. Because he studies asthma, no?
He studies
the impact of the fungi on asthma.
Yeah. And he every day he does public service that almost nobody recognizes that, but I'm going to mention it here because he counts spores in Puerto Rico to inform media, especially to inform asthmatics and people with respiratory health problems, on the types of spores, the species. He goes on the microscope every day. He has stations in Caguas, in Mayagüez, in San Juan. So with that data, so we recruit patients and we have their microbiomes, specific dates. And for those specific dates, Dr. Bolaños tells us what were the
levels.
The levels and the species of the fungi that were there. So we have a very cool paper which was published, I think, 2022 for a master's thesis of a student, where she showed that when you have periodontal disease, when it's more severe and there's more fungi in the air, the protective fungi in your oral cavity decrease by competition.
Is that right?
So your weaker immunity allows for external fungi to come and colonize, and your own fungi disappear. Because we also have a fungome.
A fungome.
That or mycome.
Mycology.
That it's understudied.
Yes, I more the microbiome, but not the fungi.
Yeah, it's more based on bacteria than fungi, and that's one of my main interests right now. It's to look at, especially vaginal microbiota and cancer progression.
So that's talk to me a little bit about these new projects that you have at your lab.
Okay, so we have been working since I was at Intermetro. So there was also a gap here.
Yes, I know.
Okay, so I made my postdoc in JGI, so it's the Department of Energy. I looked at enzymes from this flying cow to detect for enzymes that break down cellulose to produce alcohol. And then I learned a lot of metagenomics and bioinformatics there. And after the postdoc, I came to Interamericana, which is a private university in Puerto Rico, Intermetro, where I was given my first chance to be a PI. So I developed my first lab in microbiology there, and I only had undergrads. So the chancellor asked me to develop a master's degree. So for 2.5 years, more or less, me and a colleague who is now at CDC, Rafi Tosado, we developed, and other colleagues, we developed a master's degree. And I created a bunch of courses especially for people who worked at industry to be able to get a master's in microbiology and to improve their life. No? That was the main goal. And the program still runs today.
Wow.
And it has like 40 students.
Wow.
So it's a year program, master for 1 year. You have a master's degree in microbiology, but you don't have a thesis. You have a tesina, a small report. And in Intermetro, I started, developing ideas with colleagues, with Eugenio Santiago. We did tabebullas, the roble común, tabebullas in Puerto Rico, the rhizosphere, the soil with Carla Restrepo. And then I chatted out to the School of Medicine. I'd never been there. And one day I want to meet the OBGYN department. I want to meet with a gynecologist because I'm interested in HPV and about the microbiome and HPV. There are no studies in Hispanics at all, not even in the US at the time.
Wow.
And so I met this wonderful lady. She's also a mentor and a friend, Dr. Romaguera. You should invite her to the podcast. She's amazing.
She is.
So we started working back in 2014. So it's been 12 years now. And we started this big cohort of women. So I wanted to swab everyone, vaginal,
yes,
anal, saliva. And we started genotyping HPV and looking at the microbiome. And then we found a link. When women have the cervical disease, they have very few lactobacillus. And then we started publishing. The project got bigger, we got funded, and then it continued. Then Hurricane Maria came up, and we lost all the freezers because we didn't have a generator at Intermetro.
Really, Filipa?
Yeah, I lost all my I started from scratch in 2018. It was very hard. It was a hard experience.
I bet.
I only saved the DNAs who were frozen, and then I sent them to Maria Gloria, but it was like 3 weeks after the hurricane because FedEx was only for humanitarian purposes. They didn't allow us to send research samples.
Yeah.
I kept the DNAs in my freezer at home, and then in December that year, I went to NYU and I sequenced the samples, and I took my students with me to NYU. We transformed this experience in a good experience, and then we published a paper. And then I got a call from the School of Medicine. They knew that I had lost all my collections, and they didn't have anyone working on microbiome. I was, in fact, the only one in Puerto Rico doing that. And they said, why don't you come here? We don't have a starting package, but we can pay your salary. We can negotiate. And I'm like, okay. And then I went there, and I'm here. And here I am.
Yes. So that was really
life takes you. Life takes you, and you have to let yourself go.
So Filipa, we were discussing your research projects right now at your lab. Can you tell us a little bit about what are the major aims? Because I know you have several, but...
Sure. So continuing from Dr. Romaguera's project with me, which lasts for 14 years. So in that, we recruit women that come to her clinics for an annual pap smear.
Yeah.
And then we categorize them basically in healthy, normal, no lesions, no HPV, into pre-cancer and cancer, right? So we collect vaginal swabs, urine, saliva, different types of samples. So that mission, the goal of that project, I don't know if you guys know, but in Puerto Rico, despite vaccination, HPV is on the rise, and cervical cancer still is a major cause of death. So we don't know why. And our hypothesis is that the microbiome has a role in the persistence of the viruses in Hispanic women. So what we're doing is genotyping HPVs, really high throughput, We are looking at viruses, we're looking at fungi, we're looking at the host response, cytokines. We're looking at all the lifestyle. So we do a very large questionnaire about sexual behavior, health, dietary supplements, everything that you can think about. And then we have all the clinical data of the patient. Mm-hmm. So we have different projects looking at, for example, Natalia Pagan, a student in my lab now doing her PhD. She's interested in understanding, because she's finding HPV in the oral cavity of these women, and she's matching them up with the HPVs in the cervix.
I see.
And she's finding that, there's a correlation, very strong correlation between the HPVs in the cervix and in the oral cavity. And we had previous studies. I have a paper with Johns Hopkins looking at head and neck cancer. And from analysis that I did, we know through the HMP, which is the Human Microbiome Project, which is basically healthy Caucasians from the Baylor College of Medicine, like medical students. Most of that data is from there. So when we looked at the head and neck cancer patients, and I found lactobacillus. And I said, well, these people could be eating yogurt, okay. But then I wanted to look at what types of lactobacillus, and then I looked at HMP vagina, and I found a lot of vaginal samples, vaginal lactobacillus that were matching those species in the head and neck samples. So there was like an oral sex route more likely involved there. So Natalia is looking at that, sexual behavior, oral, anal, vaginal, digital, and we do several types of questions around that. And she's trying to develop a sexual risk score so that we look at the microbiomes through this lens. Then we are starting a new project. This one is led by Annelise Dominici. She was an undergrad in my lab here in environmental science, I think Department of Biology or Environmental Sciences. And she did her tesina with me looking at the microbiota of sea urchins with a colleague in Bayamón, Ruber Barreras, who was a grad student also here in biology. And she published her paper for her undergrad thesis, and she continued in the lab. She worked with the HPV project, so she understood a lot of the recruitment of the patients. And then she said, I want to do an MPH. So in her thesis for a master's degree in public health with Ana Patricia Ortiz from the Cancer Center, we wanted to look at anal HPV prevalence. And her paper now is in press. And we had the DNA genotype, so we sequenced the microbiomes, and now she's looking at anal microbiomes. But her main project, which she's really very excited to lead, is with Maria Gloria too. It's an MOU that we have with Belgium, within Antwerp University, to sample women across Puerto Rico. We plan to swab 8,000 women. It's called Project Lola. It's in honor of the first medical doctor in Puerto Rico, Dolores. There were two medical doctors. I don't remember her first name, Dolores Marchand and Dolores Piñero. What was common between them was Dolores. So I said, so this project is called Lola. So in all the countries in the world that are part of this consortia, it has the name of an important scientist or an important OBGYN or an important medical doctor. So we chose Lola because one was from Jayuya, which is rural, and another one was from San Juan. This is from the 18th century, the first two medical Puerto Rican doctors. And this study aims to look at vaginal microbiomes across a gradient of urbanization. So looking at rural areas in Puerto Rico, and we have from children to centenarians.
Wow, that's fascinating, Filipa.
And when we have our IRB protocol approved, anyone can participate. And you can share your samples, and we will give you your microbiomes, and it will be then, because it's the same methods across the world, and we can do meta-analysis and look at what is changing. And we look at the habits, the diets, the lifestyles, etc. It's a very exciting project. Then we have also a very cool collaboration with the internal medicine department to look at Alzheimer's. We had our first paper published in 2025 with Dr. Vanessa Sepúlveda. She's a geriatric specialist in Alzheimer's. And we are looking at how the gut microbiome is associated with cognitive decline. And we're looking at also controls because and this project is particularly sensitive in the fact that bringing a patient to a clinic to give you poop when he has some type of cognitive impairment, it's very hard because this person has to come. You have to organize everything so they can park. That he has to be accompanied by someone. You have to make the questionnaires, then you collect the samples. It's a very, very complex way of recruiting these patients. So this, those specimens are like gold.
Yes.
All human samples are gold, but these especially are more sensitive because of this, because it's very hard to recruit these patients. And what we're seeing is that some of the controls, so the younger people that come with these patients, have some type of cognitive impairment.
Also.
But they are not Alzheimer's patients. They had no idea they had some sort of, and sometimes and one of the things in the paper that we showed is that there's even in the control group, we have a cognitive impairment scale and the diversity of the gut decreases. Huh. So it's very interesting.
Very interesting.
And we should start, and that's the important thing that what we're doing now is educating the community about eating well, eating fibers, and getting your gut richer, because it has a bunch of effects, and that's one of the things. Cognitive impairment depends on, one of the things is not only genetics, having ApoE alleles and all that, but to have a good anti-inflammatory diet, it will certainly help. So we have that project. Also with internal medicine, we have a plan to develop a protocol with Mavi.
Okay.
Because there's also a paper coming out showing that Mavi has a lot of probiotics. I cannot disclose too much about it, but it has a bunch of probiotics. And so we want to give Mavi to patients with inflammatory bowel disease and look at what's going on.
Wow.
These are some of the things that we're doing.
Yeah, I know. You seem to be quite a busy bee, and from the standpoint of research, because you are a very highly valued scientist in Puerto Rico as well. So let's move toward one of your dreams come true, which is to create the Center for the Microbiome here now at the Cobre grant. So talk to us about the Cobre grant and what does it mean, where is the focus?
Right. So the Cobre is the Puerto Rico Center for Microbiome Sciences, started by NIGMS, which is the National Institute of General and Medical Sciences, which actually funds several important institutional fund centers in Puerto Rico, including Puerto Rico IMBRE and Alliance, which is a clinical, very big clinical facility. And there are other Cobres in Puerto Rico on neuroplasticity, on cancer, and then there's ours, which is the youngest one. So the way that it started, it coincided, it was in 2024, mid or I think fall 2024. I was approached by the editor of Trends in Microbiology, and he said, write a piece about the status of microbiome in Puerto Rico. I don't see, except from your papers, I don't see much. So I wrote like a small, not a review, but an opinion paper.
An opinion paper. Yeah.
And I basically said that we should have a center to train folks, because the limitation is not, the equipment, is not the sequencers. We have the sequencers. We have the limitation here is implementing data analysis and integrating all this data. And then in December 2024, Mark Miller, who was a professor at the time, I didn't know him personally, the pre-award officer at the medical school told him that since another neuro-type Cobre was not possible, that they could talk to me if I was interested in developing something microbiome. And I said, for sure, I have it in my head. It's my dream come true. Let's do it. And we did it together. So with Dr. Sosa from the anatomy department and Dr. Miller. So that we are the leadership core, the leaders of the Cobre. And we started this center to really, the mission is to expand microbiome sciences in Puerto Rico, give the opportunity to junior faculty and also mid-career, not only juniors. And I purposely did that because I was also, at some point I was a mid-career scientist and I was striving to I got my R21, it was like, oh, and it's very hard. After your 10-year, after you stop being an early career, it's very hard to get funds unless you have a bunch of preliminary data. So I think it's important, especially in a school of medicine where residents have to have research and medical students need to graduate with a research experience. It's important also to give the mentors, the mid-career ones, and the ones who want to change gears.
Exactly.
To have the opportunity to develop their projects. So this big COBRE award is to train students, train faculty, and fund new projects based on microbiome to advance biomedical sciences in Puerto Rico. And we're doing that. First, we started supporting 3 amazing researchers, one in my department, Dr. Stephanie Dorta, an immunologist with interest in cancer, in head and neck oral cancer. Dr. Yancy Ferrer, interested in epilepsy. And Dr. Morales, Roberto Morales, working with fear and aggressive behavior in cave fish. And they have also amazing mentors. Local mentors, and then we have a very important external advisory committee with Clarivet highly cited researchers, Dr. Jack Gilbert, Dr. Janet Jansen. She was interviewed recently by the Blueprint of Life, and Dr. Martin Blazer. And so they help us, empower these 3 faculty to have the most productive projects possible. And now we have a call for pilot projects and feasibility, which you can use core facilities to develop, to have your data analyzed, to have your data sequenced. And we want to scout the community to see what are the future RPLs, right? We want these guys to get R1s and we want new ones. So it's open to all UPR campuses. And eventually, we expect that other institutions in Puerto Rico be involved as well.
Great.
Yeah.
And I just saw that you recently had a symposia.
Yeah, we had the first microbiome symposium in the Caribbean.
Oh.
We weren't, it was free because it's part of our plan in the grant. So we wanted to do first, we didn't imagine we could have so many people there. So we did it in the same hotel we did the retreat. And at first, the day before the deadline, we had 20, 30, 40 posters, not more. The deadline, we had 85. And we're like, and then the mentors want to come in, and it's more than 100 people in the hotel. Oh my God, how are we going to do this?
Right.
So we started, being really creative with, everything. And I think it was a very cool event. We had folks talking about new bioinformatics tools from George Washington University. We had Maria Gloria came as a homage. She was the keynote on our first symposium. Then Ana Luisa came from UMass. She was also another student at Maria Gloria's lab here in Puerto Rico. And she's now an expert in IBD, IBS. And she told us about this anti-inflammatory diet that she developed. And then we had my postdoc also presented her amazing work in metabolomics, gut metabolomics. Not gut, yeah, she presented gut and some of the vaginal work she's working too. We had, I think, more than 45 PIs in Puerto Rico representing their labs, from Mayagüez, Rio Piedras, Humacao. Everybody was there. I was so excited. There were 85 posters, 200 presentations, flash talks. Yeah, and the next one is December 1st, so save your date. We don't have the place yet.
Okay.
But it's December 1st.
Wow. Well, you've been busy.
Yeah. We never stop doing cool things.
I know.
We play, right?
Yeah.
I always tell my students, we don't work, we play.
Exactly.
Because they're like, do you have a life? And I'm like, I love what I do.
Exactly.
Why should I complain?
Exactly. So that is one of the, throughout our interviews, we see the passion that all scientists show on what we do. And that really is what determines how successful we are in each of our field. So wrapping up our conversation today, which I had a lovely time. Could you talk a little bit with the Cobre and the things you've been doing of collaborations with this project of the Blueprint of Life?
Yeah. So I have the privilege to know Ricardo from years ago, and he showed me the project early on. I was involved in one of the visits from NSF to Puerto Rico as an external, let's say, evaluator. I was like moderating the meeting, and I learned a lot from the Blueprint. And I think that day we started thinking like we can do things together. We can co-host seminars, and we do. The Blueprint logo is in everywhere. So we share the evaluations from our events. We didn't have yet, because our Cobra is 7 months old. So we didn't have yet many workshops, but we intend to develop workshops together and to co-host projects together. That's totally something that we want to do. But so far our collaborations have been mostly on seminars.
Yeah, well, that sounds great, because it's open to possibilities.
Yes.
And I think that's wonderful.
Yeah.
Well, thank you, Filipa,
You're
for
welcome.
being here with us. We love to hear what you're doing, and so proud of you. So
thank
proud
you very
of you.
much.
And so thank you for being here, and we look forward to listening to all the wonderful things you have on your list of accomplishments.
Thank you.
And we look forward to seeing everyone on our next episode.
Thank you. Gracias.
Desde el Origen is supported by the U.S. National Science Foundation through the eRISE program, award number 2435987. The opinions shared here are our own and don't necessarily reflect the views of the foundation.